Okay, we did not spend as much time talking about nucleic acids. So what are the primitives of nucleic acids? Anybody know? Primitives means like the subunits, the MERs.
Phosphate backbone.
That's a piece of the MER. What's the actual MER called? Yeah, it's a nucleotide. Okay. What goes into this is called DNTPs, deoxynucleotrophosphates. So they look like this. They have 3 phosphates. There is a 5-carbon sugar that looks like this, with a base up here and oxygen here. What are the bases? What are the options? How many bases are there in DNA?
Hydrogen.
Bases?
4.
There's 4 bases. What are they? Yeah, there's A's, T's, G's, and C's. This should be review. So the difference between the A's, T's, G's, C's, this is kind of like an R group. This varies in A, T, G, C. This is the backbone. So this is the building block that gets put into DNA. Okay. So some interesting things to point out, or actually maybe we should review. If you are thinking of structures, organic chemistry structures, since some people have not had organic chemistry, what's that? If this was an organic chemistry structure? What?
It's a carbon bond to another carbon?
Yeah, this would be a CH3 bound to CH3. That's what this is. So anytime you see like a vertex like this in organic chemistry, this implies that there's a carbon. If there's nothing else attached to that vertex, it implies that the carbon is saturated with bonds either to other carbons or to hydrogens. So this carbon right here would be, that would be CH3 here on the left, and then it would be CH2, CH2, CH3. That should be review. If this is confusing, you should spend 5 minutes looking at a YouTube video explaining this. Okay. Otherwise, you won't really see what we're drawing up here. Okay, so this is a 5-carbon sugar. The carbons have numbers. This is carbon 1, 2, 3, 4, 5, hence 5-carbon sugar. Okay, what's the difference between DNA and RNA? So this right now is DNA. Why is it DNA? Why is it a DNA base?
Deoxyribose.
Yeah, deoxy. So that's what the D actually stands for. That means at carbon 2, there's no oxygen. It's just a hydrogen. Okay, so how is RNA different?
It has a ribose sugar.
Yeah, so RNA at this position has an oxygen right there. That's the difference between DNA and RNA. Okay, there's another one that's worth pointing out that you should know for biotechnology, which is called a dideoxy NTP. This is a dideoxy NTP, no oxygens. So dideoxy NTP has no oxygens. This is actually a really important reagent for biotechnology and molecular biology, because this is a very important reagent that goes into sequencing reactions, okay? If you add this base onto the end of an extending DNA molecule, what would happen if you add DDNTP at the end? What happens to that DNA molecule?
Would it just stop?
Yeah, it stops. It stops. You cannot elongate. So remember that DNA is a form of a polymer. It's just multiple bases connecting to each other. And if a DDNTP gets added onto the end, that kills the ability for the DNA to elongate, because DNA forms a chain through this oxygen. So as DNA extends, it's through a bond through that oxygen, okay? So if you get rid of that oxygen, you can't elongate the DNA chain. So remember that. This will come up later, and I'll talk more about that in sequencing. Okay, so now that you've seen that, you should also look at this figure in the notes, okay? So when these bases come together, some of this energy is removed, okay? So only one phosphate is left over when the chain elongates. So essentially this O connects to the phosphate of another base, okay? So this is how the bases connect together from here to here. And this is important to know. It's important to understand the structure because the way that the DNA connects together determines its direction, and DNA actually has direction. So when you hear of things like 5' to 3', which we're gonna discuss, the derivation of where that comes from is because the DNA is connecting, the bases are connecting from the 5th carbon to the 3rd carbon. So that's where the direction of DNA comes from in the actual molecule. Any questions about that? Okay. So this I talked about yesterday. All DNA chains have phosphates, and what charge is phosphate?
-3.
Yeah, I don't know about the number, but it's minus, yeah. It's minus, okay, it's negative. And so all DNA by its nature is negative, like entirely negative. It's very negative, okay? That's the negative phosphate backbone of DNA. Good. Okay, final thing about DNA, maybe not the final thing. So the bases come together and pair in what's called the double helix. So DNA is double-stranded. So if you see DS DNA, that means double-stranded. It's double-stranded. The bases are binding to each other. This is one phosphate backbone. This is another phosphate backbone. How do the bases come together? Which binds with what?
I always get it wrong. It's like A equals T is equals G, but I always forget like where they actually are.
Okay, don't forget that anymore, although you got it right. Okay, so A binds to T, G binds to C. So just remember that. That's easy to remember. A, T, G, C. A's bind together with T, G's bind together with C. How many hydrogen bonds are connecting these bases?
Doesn't G and C have 3?
Yes, and that's this figure in the handouts here. Okay, you need to know that. This is very important. This will for sure be on the test, and it's super simple. So it's like, okay, just learn it. A's and T's have 2 hydrogen bonds that are holding the chains together. So in here, if this was an A and T, there'd be 2 hydrogen bonds holding the chain together. G's and C's have 3, okay? So if this was a G and C, there are 3 hydrogen bonds holding that chain together. So now let me ask a question which is important for biotechnology, because this becomes important for PCR design and PCR primers. Which one of these is stronger? Which one is a stronger bond?
G and the C.
The G and the C. So which one is going to be easier to break?
A and the T.
A and the T. Yeah. So when you do PCR, you break apart the DNA in denature steps, and you also send in primers that anneal, and the temperature at which those primers anneal is determined by the AT or the GC content, because that determines the strength of the bond of your primer to the DNA backbone. Okay? So this becomes actually extremely important, knowing that A's and T's are a weaker bond and G's and C's are a tighter bond. Any questions about that? Okay, so that's just a little bit on DNA. Now I have to see where we were actually at. Okay. So DNA has direction. So it's more accurate to think of DNA as kind of like a directionally oriented molecule. Okay? So you often write it like this. 5 to 3, 3 to 5, okay? This is important because the enzymes that interact with DNA can only interact in certain directions. It's like a left hand and a right hand, okay? So if you see the term sense, what does sense mean? What direction is sense?
5 to 3.
Correct. So sense, the sense strand is the 5 to 3 strand. The sense strand, the reason you call it sense is because it makes sense to the ribosome. So messenger RNA is also in sense orientation. So messenger RNA is always in what direction? 5 to 3. That means messenger RNA is made from this bottom strand. It's produced 5 to 3. Okay, so the ribosome can only read 5 to 3. It can only translate 5 to 3. So you have to, when I first started learning this was difficult, but it becomes really important. So I kind of have like a simple way to kind of like help remember this. I think it's helpful to me. So when I think about like direction, I just have this like mnemonic trick where I draw a triangle and I think like this is higher than that, so I'll put the 5 up here, put the 3 up here, okay? And when I think of like a ribosome, I think of something like big and fat. And it's hard for big and fat things to kind of like climb up hills. So big and fat things, it's easier for them to like roll down. This is all kind of just dumb, but this is how I remember, okay? So if you think of like a ribosome, it's much easier for it to roll down the hill. So ribosome will read 5 to 3 prime because it's like rolling down the hill. Okay. The other type of protein we're going to talk about today is polymerases. Polymerases I think of as kind of like a smaller, more agile thing, and they can read uphill. So polymerases read 3 to 5, and they write 5 to 3, okay? So remember that the ribosome reads 5 to 3, but polymerases read 3 to 5, and they write 5 to 3. That's how I remember that.
Does read and write mean?
We're gonna talk about that, good.
Okay.
So that's literally the next thing we're gonna talk about, good. Okay, so let's talk now about DNA replication. Okay. So we're still actually not to the central dogma yet, so just keep that in mind. So DNA replication. DNA has to be replicated in the cell. Why is DNA replicated first?
Make copies.
Why would you make copies?
Doesn't the DNA degrade? So you need more.
Okay, so that's an interesting question. Okay, let's first address that. You're kind of implying that cells have multiple copies of DNA, which is kind of true and kind of not true. So let's just quickly like talk about this. If you have a bacteria, how many copies of DNA does the bacteria typically have of its chromosome? Yeah, it typically only has 1 copy. I mean, there's some biology is always gray. So like when does the bacteria actually have 2 copies?
Right before mitosis.
Yeah, right before it's about to divide. Okay, so the first reason of like why you replicate the DNA is like if you want to divide the cell, if you want to replicate, the other cell needs a copy of the DNA. There are cells that have multiple copies, kind of like what you were applying. Which cells have multiple copies? Give me one example of one cell that has multiple copies of DNA. Humans. Okay, good. I'm glad we got that. Humans have 2 copies. If you have 2 copies, what's that called? Well, okay, but it's typically given a name of diploid. Versus if you have 1 copy, that's haploid. So this is just stuff to think about, okay? But the real reason like the cell is replicating DNA is because the cell needs to replicate itself, and the other cell, the daughter cell, needs a copy of the DNA. Otherwise, it would not persist. So we're going to talk through how DNA replicates, okay? So we should have all seen this before. I'll draw it closer together. But now let's go through it, this DNA replication, and let's actually learn the players of the cell, and let's memorize their names, and let's learn the steps of DNA replication. So here's a DNA molecule. Let's do, I always draw this wrong, but hopefully I'm gonna do it right. Okay, so here's the DNA. What's the first step of replication? What has to happen?
You only need one strand.
Yeah.
So you need to have an enzyme break apart the helix.
Yeah, so that has a specific name. I wouldn't call it breaking, because that makes it seem like it's breaking.
Or like separate.
Yeah, separating. That's a better word. There's actually a molecular term. Does anybody know the molecular term?
Annealing.
What'd you say?
Annealing.
Annealing is when something comes in and closes up. It's the opposite of annealing. Do you know?
It's on the tip of my tongue.
What does it start with? Oh, somebody said it. Denaturing. Okay, so that's the first step. So the first step of replication is denaturing. Okay. So how does the cell denature the DNA?
Enzymes.
Yes, there's an enzyme. Okay. What's the enzyme's name?
Helicase.
Helicase. Okay, so this we're now starting to learn the players of the cell. These will be on the test. If I tell you the name of an enzyme, that means I think it's important enough to teach it. Okay. So you should now know the name. And never forget the name of helicase. Okay. So helicase is an enzyme that does the first step of replication. Helicase comes in and it creates essentially, it opens up the DNA through enzymatic processes and creates this replication fork. Okay. So now we have this after helicase. What's the next step of replication? Not elongation, not yet. I think you were about to say it right. What?
I don't know.
I think you were actually about to say it right. What do you think should happen?
Something's gonna recognize it.
Something's going to recognize it. Okay. So the first thing you need to know is for the enzyme that really needs to recognize it, it's hard to grab onto a single strand. So what might it need to help it like grab on?
A
A
primer.
primer, kind of, yeah. So there's an enzyme called, what do you think it's called?
Primase.
DNA primase, okay. So there's an enzyme called DNA primase, okay. And the job of primase is to create a primer. So DNA primase comes on and creates a little primer. See, here's where I always get direction confused. Let's see. It's gonna read. So I'm gonna switch these numbers. Imagine this is the 3 to 5, and this is the 5 to 3. So DNA primase creates a primer. That primer is 5 to 3. Okay, so DNA primase creates a little oligo. And oligo is a sequence of base pairs strung together. That's a short sequence. Okay, that's what oligo means. So it creates a little oligo, a little primer. Okay, that's the second step. Priming, or perhaps you might call this annealing. Okay, what's the third step? Somebody said it before. Now's your big chance.
Extension.
Extension. Okay. What extends? What is the enzyme that does the extension? So remember, this is a good example of how proteins are the verbs in the cell. What's the protein that does the extension?
Ligase.
Nope, not ligase. Not there yet.
DNA polymerase.
Yeah, polymerase. So let's do purple. And I just showed you the little mnemonic. Polymerase, which direction do they read? Yeah, okay, so we're learning the mnemonic. They can read 3 to 5. So that means this is 3. That means they can read this top fork going this way, okay? So polymerase comes on here. It reads 3 to 5, so it's reading the 3 to 5 strand direction, but as it's extending, as it's putting on bases, it's writing, which this is what I mean by writing, it's writing in what direction? 5 to 3, yes. Okay, so now we understand the direction. This is important to know because polymerase can only go in that direction, can't go the other direction, okay? So on this strand, what's this strand called? If this is what's called the leading strand, yep, this is a leading strand because the polymerase can just keep reading and keep writing in this way. Whereas this one is the lagging strand because the directionality is problematic. It can only read this way, so it's moving into the fork, okay? And so you get these primers, and the extension fills in these gaps. There's a name for these? Good. We have good background. Okazaki fragments. Okay. And this just persists. So we have the leading strand, just writing, the lagging strand, making these fragments. What's the final thing that has to happen? So this is now replicating the DNA. This strand is essentially okay. What's the problem with the lagging strand?
Has to be glued together.
Yeah, it's chunkified. It's in chunks. So what needs to actually be glued together now that we just drew the structure of DNA? What's the actual molecule that needs to be glued together? What needs to be sealed? What?
Was it the phosphate backbone?
Yes. So what we say is that the phosphate backbone has to be sealed, and there's an enzyme that does that. Now somebody else said it before. What's the big enzyme that does that? Ligase. So the ligase. So we've learned polymerase. So we've learned helicase, primase, polymerase, ligase. Ligase seals the phosphate backbone. Okay. So ligase will come into these spots and make it one polymer.
And that's only for the lagging one?
Yes.
Does ligase do it for the leading one as well?
I'm pretty sure it's only the lagging,
Okay.
but fact check me on that. Okay, does that make sense? Any questions about that? This should be review. But what you should now never forget is the names of the enzymes. Because in biotechnology, or molecular biology, or synthetic biology, we often want to replicate DNA ourselves, right? So oftentimes the way that molecular biologists do it is often the way that the cell does it, with slight modifications. So what is it called when you replicate DNA in a tube?
Is it PCR?
Yeah, PCR, which stands for what?
Polymerase chain reaction.
Okay, good. Okay, so the steps of PCR are the same, essentially the same as in the cell, just only slightly different, okay? So the first step of PCR then should be what? So if you have a molecule of DNA, what's the first step of PCR?
Denature.
Yep, denature. In PCR, do you use helicase? What do you use?
Nothing.
Heat. Yeah, so in PCR, the denature is not done enzymatically, it's done with heat in what's called a thermocycler. So the thermocycler heats up, okay? Eventually we'll go over the program. Let me just remember that. So you heat it up, that denatures the DNA, splits it apart. Second step is what?
Annealing.
Yep. Do we use DNA primase in a PCR reaction?
Use
No.
DNTPs.
Do you use DNTPs? Why is that? Why is it wrong to say that you use DNTPs to prime?
Oh, no, not as a primer.
Yeah. So what do you use? What do you use to anneal? What
A
anneals?
specific primer?
A specific primer. Yes. So you have a primer. You have a forward primer. So a forward primer is typically in the 5 to 3. So if this on the top, if this was 3 to 5, your forward primer, 5 to 3 direction. You have a reverse primer. So there's two primers. Reverse primer is the we'll deal with that in a second. If this is 5, 3, this is 5, 3. So your reverse primer is technically build 3 to 5. Okay, so we can see that. So you build these little primers. About how large are these primers? Who's done PCR?
It's about 15 base pairs.
A little bit more than that. It's usually, you could probably do a PCR with 15 base pairs, but usually I think it's about 24. So usually, just to get a sense of size, so these are also called Legos. They're about 24 base pairs, about. Okay, so we have annealing. What temperature is sufficient to denature DNA? So how hot does it have to be?
Depends on how long the strand is.
Yes, but only to an extent. Like there's a certain temperature that will denature everything.
Around 72 degrees Celsius.
72 is not enough. Well, typically you denature at 98. So when you run a PCR reaction, typically you're denaturing at 98 degrees Celsius. So you're almost boiling, essentially boiling the water, okay? Annealing temperature is gonna vary based on when these primers will stick. And then we have, what's the third step?
That's extension.
Extension. Okay, and extension now usually runs at, somebody said it, extension actually runs at 72 degrees Celsius, depending on the enzyme, but typically 72. Okay, so what enzyme extends? What enzyme are you actually going to use in a PCR reaction?
Polymerase.
Yeah, it's going to be a polymerase. Okay, so your polymerase will come on. They're going to read 3 to 5, and it's going to write. Okay. In the first reaction, you'll have double. Then in the next cycle, so then you initiate a loop. You cycle through this, and each time you get a 2x, twofold increase, which then essentially is an exponential curve of copying the DNA. Okay, so that's PCR. Are there any questions about that? This is like the most simple molecular biology reaction. So now let's go a little bit deeper. Let's do, how much time do we have left?
30.
Okay, we got plenty of time. We got plenty of time.
20.
What?
I added it to an hour. I'm sorry.
So how much time do we have left?
20.
20 minutes. Okay, that's still plenty of time. Okay, so let's do the program, and let's do the recipe for PCR. We should know these and never forget these. Let's first do the recipe. So if you're going to do a PCR reaction, what goes into the tube? What goes in the tube?
DNTPs.
DNTPs. So we just learned that the primitives of DNA are DNTPs. So if you're going to write DNA in a test tube, you need DNTPs. So you need A's, T's, T's, and C's. A mixture of DNTPs, okay? What else do we need?
forward and reverse primers.
Yep, 2 primers, forward and reverse primers. You design these. I'll teach you how to design these, but not right now. So you need forward and reverse primers. What else do we need?
Polymerase.
Polymerase.
Attack polymerase.
Let's talk about that. So, when Carey Mullis first invented PCR, he did use Taq polymerase. What was the rationale for using Taq polymerase? What is Taq and where does Taq come from?
There was a quote, it came from a hot spring, and it can stand hot temperatures.
Yes, so that was the rationale for using Taq. Is in this process, in order to denature, we're heating it, and so you need an enzyme that is going to be stable through high temperatures. Okay, so that's why you use Taq polymerase. But most people do not actually use Taq polymerase anymore. Taq polymerase is kind of a crappy polymerase. So here's kind of like some new knowledge that hopefully you'll learn. All polymerases, so enzymes have properties. Okay, so there's what's called an error rate, and there's a speed at which they can write. Okay, Taq polymerase is actually really bad. Taq polymerase has an error rate of 1 in 1,000. So if you're writing a copy of DNA that's 1,000 base pairs long, you'll get at least 1 error every replication. If it's 10,000 base pairs, which would be a real long PCR, you're going to get 10 errors every time, and that's going to exponentially be pretty bad. Okay. On top of that, the speed is not very high. I don't remember off the top of my head the speed, but essentially what you can remember is it's slow. Okay. So there's actually nowadays we don't really use this. We use it for certain types. Like if you're cheap, if you have a cheap reaction, you don't want to spend a ton of money. So that's the one benefit of Taq, is it's cheap. Okay. If you're doing kind of like a reaction, you don't need perfect fidelity. You can just use Taq polymerase and you'll save some money. So that's maybe a good thing about it. But nowadays we have what are called high fidelity polymerases. So one of these, here's 2 new names. One of these is called Fusion. Another one of these is called Q5. Most of the time in a molecular biology lab, if you run a PCR reaction and it's important, you'll be using one of these. And because the high fidelity ones have a 10x increase in fidelity, so they have an error rate of 1 in 10,000, okay? And these ones are fast. You can typically write 1,000 base pairs per 15 seconds. These are a little bit more expensive, but they're actually not so bad. Okay, so keep that in mind when you're doing your recipe, is that Taq polymerase is kind of like, you would only use that in certain situations where you're trying to save money. Okay, so what else goes in our PCR reaction?
For this specific to polymer, that polymerase uses.
Yes, a buffer. All enzymes come from biological organisms in the cells, and the cells have a specific concentration of ions. So those polymerases are evolved to work only in certain ionic conditions. So there's always going to be a buffer. There's typically going to be magnesium in that buffer. What else goes in their reactions?
You need a template strand or cDNA.
Yes, very good. So we need our template. That's what it's binding to, so some kind of DNA extraction. And then the final thing, which is something simple, don't overthink it.
It's water.
Yeah, water. So you're gonna kind of like, whatever total volume you want, you fill the rest with water. What kind of water would you use?
Distilled.
I would not use distilled water. So there's grades of water. There's kind of like tap water, there's distilled water, and then there's called like molecular biology water, which is essentially like RNAs free
We're using DI.
and DNA is free. Well, distilled, deionized. So there's kind of like distilled, deionized. These are still not like molecular grade water. So actually, if you're gonna run a PCR reaction, don't put any of these. These are kind of like shitty waters. There's specific water you want, which is not gonna destroy your nucleic acids. Use the high quality water, okay? So that is what goes into PCR reaction 2, okay? The program, typically there is initial denature. I'm gonna erase this so I have more room. Hopefully everybody got that. There's typically initial denature, which is about 2 minutes. at 98 degrees Celsius. There is then the initiation of your loop. So the first step, denature, anneal, extension. So denature is typically 98 degrees Celsius, 10 seconds. Anneal is typically about 56 degrees Celsius. We'll talk more about this for 30 seconds. Extension is 72 degrees Celsius for a certain amount of time, depending on how long your product is. Maybe let's say 60 seconds. Could be faster, could be slower. How many times do you usually cycle this?
36.
Yeah, sometime about 35 to 40 is normal, okay? And then there's usually a final extension, which usually runs about 5 minutes at 72. And then you hold it on cold for infinity until you're done. So here's a typical PCR thermocycler program if you've never seen one. Hopefully this all makes sense, okay? All right, let's move to the next thing. So now let's actually get to central dogma. How much time do we have left now to discuss central dogma? 14 minutes, okay, good. So what is the central dogma of biology?
DNA to RNA protein.
Okay, so let's just draw quickly the central dogma. DNA to RNA to protein, okay? What's this process called?
Transcription.
And what enzyme does transcription? What's the name of the enzyme that does transcription?
Transcriptase.
Okay, yeah, yes, but it's also a type of enzyme that we've already talked about.
RNA polymerase.
Yes, it's an RNA polymerase. So there's an RNA polymerase, which is different from a DNA polymerase. So a DNA dependent DNA polymerase will do DNA replication. A DNA dependent RNA polymerase will take DNA and make RNA. Okay, so transcriptase is a DNA dependent RNA polymerase. Okay. This step is called what?
Translation.
Translation. Don't ever anymore confuse translation with transcription. That'll drive me nuts. What does translation?
Ribosomes.
The ribosomes. Okay. So this is the central dogma of biology. Don't ever forget this. Now your whole mind should be framed, it should be written on your hearts. The hearts of biologists. Never forget this. Okay. There's one unique enzyme that breaks the central dogma that can go like this. What's that enzyme called?
Reverse transcriptase.
Yep, reverse transcriptase. So reverse transcriptase is uniquely an RNA-dependent DNA polymerase. Where do you find reverse transcriptases? You don't answer. Where do you find reverse transcriptases? Where do they come from? It's kind of unique.
Is it in the bacteria that have RNA instead of DNA that go?
You would find them in bacteria, but not because of the reason not because they're bacterial. What type of thing has reverse transcriptase?
In viruses?
Yes. So you find reverse transcriptase in viruses, and you also find them in retrotransposons, which we'll talk about later. Okay. So just kind of like put that in the back of your brain. This is actually an important biotechnological enzyme, because what might you use a reverse transcriptase for in molecular biology? So this is a common enzyme that we use in reactions. What could you use it for?
So you know the DNA of the protein?
So you know the DNA of the protein. You could just take a protein and you could kind of like reverse translate it with a computer algorithm. So you wouldn't necessarily need a reverse transcriptase. What would you actually use a reverse transcriptase to study? Yeah, so reverse transcriptase is used as a biotechnological tool to study RNA, because RNA is less stable. Why is RNA less stable?
Because it's single-stranded.
It's single-stranded. So RNA is always, or not always, but typically single-stranded. Okay? Messenger RNA is always single-stranded. And so it's less stable. And so reverse transcriptase actually kind of like converts it to cDNA, we'll get to that later, but it makes it a more stable molecule, and so it's used to actually study RNA. So that's kind of an important thing, enzyme, to remember. Okay, so we've covered central dogma. Let's now talk about the structure of genes. Okay. So if I actually ask this in interview questions, I say, what is the structure of a gene? What would you answer?
Introns and exons.
Okay, that's good. That's a good answer, because you're starting to realize there are things inside of genes. Okay, so the symbol for a gene is typically an arrow. So whenever I draw an arrow from now on in this class, it means I'm drawing a gene. Okay, genes are always annotated typically in sense orientation, which means 5', 3'. As was said, genes often have introns and exons. Let's call this exon 1, exon 2, exon 3. What types of genes have introns and exons? Do all genes have introns and exons? Okay, so this is something important to remember, is that exons, they're splicing. So when this gets made into messenger RNA, so if I draw something like this is now a code for messenger RNA that corresponds to said gene, okay? Then splicing occurs. Splicing splices the exons together. And you can remember now for the rest of your life that exons exit the nucleus. So exons exit the nucleus, okay? So, but this is a phenomenon, splicing is a phenomenon that only happens in what types of genes?
Eukaryotic.
Eukaryotic genes. So our whole next lecture is eukaryotes versus prokaryotes. So there are some genes that don't have introns and exons. Those genes come from what types of organisms? Bacteria. So bacteria don't have introns and exons. They just have genes where the gene is actually the messenger RNA, the code, okay? So depending on if it's eukaryotic or prokaryotic, the structure of the gene might look different, but exons, introns, those are things you want to know. What else goes into the structure of a gene? So think about transcription here, the second step of the central dogma. How does transcription know where to start?
The promoter sequence.
So there's a promoter sequence. Where would the promoter be, here or here?
The 5'.
So promoter sequence is in the 5'. The symbol for a promoter looks like this, okay? So this, I'm teaching you like the molecular biology, like code language. This is what the symbol of a promoter is, okay? What else is, or what is a part of or close to the promoter? Promoters have like little pieces. Start codon would be right here. So that's in the gene, not in the promoter. So the promoter is before the gene, start codon begins the gene. Promoters have different types of regions. Does anybody know what they are?
Is it the data box?
Yep, there's a data box. There's a -35 region. Okay? What do these regions do? What's actually happening? How is the gene actually transcribed in the central dogma? What happens? What does the promoter actually do?
Help the RNA polymerase find where it's supposed
Yes,
to be.
so actually the job of the promoter is to encode a sequence of DNA that is attractive to the RNA polymerase. So the RNA polymerase will often be attracted to this site and grab onto it and start transcription, okay? That's often what happens. Oftentimes there's other things that are called transcription factors. Transcription factors, TFs, I'll write that a lot in this class. Transcription factors often bind the promoter. And so sometimes the attraction of the RNA polymerase happens intermediately through some transcription factor that's holding on to the promoter and what's called recruiting RNA polymerase, okay? So either RNA polymerase is being attracted to the promoter itself, or it's being attracted through an intermediary and then causing transcription of messenger RNA. So if you're the RNA polymerase, you know where to start because of the promoter, okay? So transcription actually starts a little bit before. So this region of the transcript, what's this region called? This has a name before the gene actually starts inside of the transcript. There might be some kind of thing about that that I don't know, but typically this is called the 5' UTR, which stands for untranslated region. So it's like the untranslated region upstream of the gene. So also too, this is called upstream. This is called what? Downstream. So 3' end is downstream, 5' end is upstream, okay? Transcripts, does transcription stop? What's here? What's right there?
The terminator.
Nope, nope. You're close, but what's, I mean, like literally like right there.
The stop codon.
Stop codon. So what are our options for stop codon?
I know there's three, but.
There's 3.
A,
In DNA, give me DNA sequence, not RNA. TAA, yep. Don't forget these. Okay, the symbol for these is an asterisk. So there's a stop codon. Does the transcript, does the RNA polymerase stop at the stop codon?
No, it has the poly A tail.
Yeah, so transcriptase, so RNA polymerase, it does not stop at the stop codon. It keeps going, okay? And there's various mechanisms that tell RNA polymerase when to stop. In general, there's a structure coded into the DNA here called what?
The terminator.
The terminator. There's typically a terminator which encodes the process of stopping transcription, okay? So this region then of RNA, what's this region called? Somebody said it before.
Poly A tail.
So poly A tail comes later in eukaryotes. A poly A tail is added on later, only in eukaryotes, not in prokaryotes. But typically this region after the stop codon is the 3' untranslated region, okay? So when the ribosome reads this, it's starting here, but it's not translating anything until it sees that start codon. And then your protein is being translated to the stop codon. So make sure you kind of understand this. Also too, there's what are called enhancers. Enhancers are further upstream and they can regulate the promoter. So the promoter is like a light switch that can control on and off. If a gene is on, what does that mean?
It's active.
Which means what?
It's being made.
It's being made into what?
Protein.
Yes, which involves the first step of the central dogma, not the final step of central dogma.
So it's going to RNA.
Yes. So being quote unquote, a gene being quote unquote on means transcription. Transcription is active. A gene being off means there's no transcription, okay? Any questions about the structures of genes? How much time do we have left now?
2 minutes.
2 minutes. Okay. What's worth going over? Final things here. When translation happens, the first amino acid is always what?
Methionine.
Methionine. In some bacteria, it can be a valine, which is a slight subtle variation of ATG. But 99% of the time, the start codon is a methionine. I'm just seeing if there's anything else worth talking about. Are there any questions about any of this? I will not ask you about mechanisms of transcription and termination, but if you're curious, you can look in the handout. All right, that's sufficient for today.